wanted to start my talk by acknowledging that I'm speaking to you and have done this research on the ancestral lands of the Coast Salish people, including the Tsleil-Waututh, Coquitlam, Squamish, and Musqueam nations. I'd also like to thank my co-authors on this work, Sally Otto and Matthew Keeley, and this is published in these two papers. If what I say today doesn't make any sense, you can go look there and see if it makes any more sense. So as evolutionary biologists, we're inherently interested in processes that generate the amazing biological diversity in the natural world. And this is from biological diversity at the species scale to diversity at single loci. So today we're going to zoom in and talk about genetic variation, this basic level of diversity in the genome. So there's a lot of standard processes that are known to impact genetic variation. And I'm going to just talk about a few of those right off that are going to come back and be important later on in this talk. So first of all, a couple of processes that maintain variation. One of those is overdominance. So this is a situation where, for example, you have a diploid locus and the heterozygote has a higher fitness than both of the homozygotes. And in this case, independent of where you start, you're in different initial conditions, your system will approach some intermediate allele frequency. And as a result of this, the genetic variation in the population, and today we're going to measure genetic variation by heterozygosity, and I'll talk a little bit more about what heterozygosity is, but heterozygosity will increase and in the parameter conditions that I'm showing here to some maximal value. But there's other processes that maintain variation, one of which is negative frequency dependent selection. So in this case, the fitness of the different genotypes changes depending on the current allele frequency in the population, in particular to favor whichever allele is rare. So if the little a allele is rare, then there is an abundance of selection favoring the little a homozygote and vice versa. And the dynamics are quite similar to what we saw in the previous slide. This is not a mistake. It just is very similar plot that independent of where you start, you end up approaching some intermediate allele frequency and you have an increase in heterozygosity to some high level. But there's also processes that deplete genetic variation, one of which is directional selection. So in this case, one of the homozygotes is favored more than the other, and this will tend to erode genetic variation and lead to the fixation of the favored allele. So in this case, you end up running off to one of the edges of this plot, and heterozygosity decreases over time until there's only one allele in the population. Another important process that's going to come up in this talk that depletes genetic variation is not a selective process, but random genetic drift. So the random sampling of gametes leads to this variation in allele frequency over time. And over time, one allele or the other allele will start to fix. And you can see that independent of where you start, the effect of heterozygosity is that, the effect on heterozygosity is that genetic drift erodes heterozygosity until it reaches zero. So I'm here to talk about eco-evolutionary processes that impact genetic variation. So one of the first ecological processes that we can think about that has the potential to maintain genetic variation is coevolution between different species. So this comes from a long tradition of the red-queen hypothesis, and as first stated by JBS Haldane in 1949, parasitism is thought to maintain favor polymorphism and increase heterozygosity because it gives a selective advantage to host genotypes in the population that are rare. So if a host is rare, then perhaps it is more likely to escape infection by the current dominant parasite in the system, and this would give it a selective advantage. So this is thought of as one of the tenets of the red queen hypothesis and has been perpetuated in the literature and can be found in modern day textbooks on evolution. Where does JBS Haldane base his ideas about the maintenance of polymorphism by parasitism? Well, let's formalize this with the matching alleles model. So in this model, you have two different host types. You have a big A host and a little a host, and you have two different parasites, a big A parasite and a little a parasite. And if you are like host and like parasite, so a matching host and a matching parasite, the infection is likely to occur due to some lock and key type molecular mechanism. And so infection occurs at this rate X, whereas if you're a mismatching host and parasite, then infection occurs at some reduced rate Y. So here's the representation as an infection matrix, and then we can write a simple system of differential equations that captures the dynamics of the allele frequencies in the system. And as you might expect, these allele ODEs for the allele frequencies break down into the traditional components of having the genetic variation times the selection. And here selection depends on the allele frequency in the other species. So if I'm looking at the host, this is the parasite and vice versa. So this can be thought of as interspecific negative frequency dependent selection. So maybe just like negative frequency dependent selection that we saw a few slides ago, this interspecific negative frequency dependent selection should maintain genetic variation. If I were to numerically integrate these differential equations, what I would observe is fluctuations in the host and parasite allele frequency through time. And these are neutrally stable cycles, so they will neither cycle out or in. They will stay exactly where they are into infinitum. All right. So right away, we can notice that there is a problem here. So here is just those cycles again, but at a different initial condition. And if I measure heterozygosity, or specifically expected heterozygosity, the probability that if I draw two alleles from this population at random, that those alleles are different, that expected heterozygosity, if I quantify it through time, and in particular, I'm going to focus on the host heterozygosity, and I calculate the average heterozygosity in the population, the time average heterozygosity, I notice that it fluctuates up and down, but over time, it approaches a stable equilibrium state. And in particular, it approaches a state that is submaximal. So despite the fact that you are cycling around an allele frequency of a half, that is the highest amount of heterozygosity that you could possibly have, this average heterozygosity in the population never reaches a half. So this begs the question, does interspecific negative frequency dependent selection actually maintain genetic variation? And in particular, we need to ask a very specific statistical hypothesis that has a null hypothesis in it, where we want to ask, does negative frequency dependent selection maintain more genetic variation than expected under neutral genetic drift? All right, so to study this, we need to build a model of matching alleles coevolution that isn't a finite population, so that it has genetic drift in it that we can compare to a model of genetic drift in the absence of the matching alleles model. So in order to do this, we have a host population and a parasite population, and then we build a Moran model so that every birth event corresponds to a death event and vice versa. So we have three different processes that create birth and death events. So you can have an infection event, and this infection occurs according to that matching alleles model. And then once an infection occurs, this leads to host death, and then a corresponding random birth event so that the population stays constant, and a parasite birth and corresponding random parasite death that leads to a constant parasite population size. In addition to these two, we wanted to include random host turnover and random parasite turnover in order to include dynamics of genetic variation that were independent of the infection process. So here I have this random host death and the random parasite death. And by changing the relative size of the infection rate versus these random turnover events, I can scale the host generation time relative to the infection rate. the net result that we get from this is a set of differential equations that look very much like what we had before. So we have the genetic variance in purple, this negative frequency dependent selection, interspecific negative frequency dependent selection in red. But now in the denominator, we have this term that standardizes the time scale to be in terms of host generations. So even though we use the word generation all the time in evolution, exactly what we mean by this can change from model to model. So what do I mean by a generation here? By a generation, I mean the occurrence of n host deaths. So as soon as n events have happened, then that is a generation in my Moran model. All right. So what happens? Well, first, let's look at the deterministic model. We've already talked about this a bit, but I just want to go over it again, that in the limit as n goes to infinity, you get neutrally stable allele frequency cycles in both the host and the parasite. Exactly what these cycles look like depend on what the initial conditions are. And then if we calculate this heterozygosity over time, it cycles to some sub-maximal equilibrium. But we can look at these allele frequencies in a different way. We can look at them in the phase plane. So on the x-axis here, we have the host allele frequency, and on the y-axis, the parasite allele frequency. Shown in red are the equilibria of this model. So the 4 boundary equilibria and 1 polymorphic equilibria, where both species are polymorphic. And this interior polymorphic equilibria is characterized by a leading eigenvalue that has a 0 real part. So this is what I mean by neutrally stable cycles, that you're on this knife edge between being a stable cycle and being an unstable cycle. And as we'll see in the rest of this talk, the fact that you're on this knife edge makes the matching alleles model particularly unwieldy because it's very sensitive to any other types of assumptions or perturbations that you can make to the model. All right, so that's the deterministic dynamics. But we're interested in comparing heterozygosity to drift. So let's talk about what happens in the stochastic world. So in a finite population, I can write down the neutral expectation for what I would expect heterozygosity to be in the absence of coevolution. And then I can run individual-based simulations and compare the observed heterozygosity in these simulations to this neutral expectation. And what we see is that early on, there is some perturbation from the deviation from this neutral expectation. So in the plot that I've shown you for the particular parameters I've simulated here, this perturbation is above the neutral expectation. And then once you're down here, you actually see a deviation below the neutral expectation. So what's going on here? This is not what we may have expected given Haldane's hypothesis. So first of all, let's talk about this deviation above the neutral expectation. To study this, we developed ensemble moment approximations for the dynamics. What do I mean by an ensemble moment approximation? Well, this is closely related to a diffusion approximation. So what we're going to do is we're going to model the diffusion of alleles in this, or diffusion of stochastic dynamics in this allele frequency space as a Gaussian distribution around this central equilibrium. So every generation, this Gaussian distribution grows, and then the ensemble moment approximation is a set of ordinary differential equations that calculate the moments of the diffusion approximation. So what is easier about these ensemble moments is that we don't need to have a solution that fits for all allele frequency space in all time. So a multidimensional diffusion approximation can be particularly unwieldy, and in particular, in this case, I can't even derive it to begin with, but I can obtain the set of ordinary differential equations that give me these moments. So if I do this, what do these ordinary differential equations tell me? Well, they tell me that this deviation early on from the neutral expectation is the result of what kind of events happen first in my model. So let's think about the different events that can happen. I can either have an infection event happen first, a neutral host death event, this is that turnover in the host, or I can have turnover in the parasite. And depending on which one of these that happens, there's some
you start at this equilibrium point, and then there's some perturbation away from that, and then selection is given by these red arrows, and selection will start pulling you in those red queen cycles. But depending on what this perturbation is to begin with, that initial effect of selection can either be to increase heterozygosity, or it can be to decrease heterozygosity. So if this red arrow pushes you into a green box, that means that event increases heterozygosity. And if it pushes you into if it pushes you into a red box, that means it decreases heterozygosity. And if it pushes you into a green box, that means that it increases it. So infection, on average, doesn't do anything. It pushes you in all sorts of directions, but no particular direction at all. And so it has no effect. But if there's a lot of host death events, this will tend to push you into green boxes early on versus red boxes, and it will lead to a slight increase in heterozygosity. So that's what we're seeing here, is an overabundance of host death events. So I can see this more generally by looking across parameter space, and what I see is that the relative rates of host versus parasite death determines this initial perturbation from this neutral expectation. So in particular, we're interested in the right-hand side of this plot over here, because that is when a parasite generation is shorter than a host generation, which is typically what we think is the case in the real world. And in that case, this initial perturbation will actually be to decrease heterozygosity below the neutral expectation. All right, so that was the early dynamics. What about this later decrease in heterozygosity? Well, in order to understand what's going on there, we have to go back to the nuts and bolts of the matching alleles model. So this is what the original model looks like. But as time goes on, drift has been occurring, and we have to think about what would happen if different alleles were to fix in the population. So what would happen if one of the pathogen alleles or parasite types were to fix? So for example, let's get rid of that little a parasite. And then what was once this really symmetric model that would lead to these negative frequency dependent neutrally stable cycles suddenly turns into a model of directional selection, where you have strong amounts of infection disfavoring the big A allele and weaker amounts of infection leading to a favoring of the little A allele. So we can see this more generally in this plot, or you can just look behind me at this quilt that my mom made of this plot that is in gray. We have ongoing coevolution, where you have both host types around and both parasite types around. But at some point, one of those parasites is going to fix, and you're going to lead to directional selection, shown here in red trajectories, and finally the fixation of one of the host types. So we can see this play out across parameter space. If I run a whole bunch of individual-based simulations, I can compare the neutral heterozygosity to the observed heterozygosity. And if I were to fall in this upper quadrant or upper triangle up here, that would be what we would expect under the maintenance of genetic variation. But instead, I see that most points fall down here in the lower triangle. And in fact, the average is a net decrease in heterozygosity. And we can see those two impacts that we mentioned earlier play out across parameter space, so that if you are over here on the left-hand side of the parameter space where host generations are longer than parasite generations, this will lead to a decrease in genetic variation. And if I take all of my simulations for a particular population size, so each of these different colors is a different population size, that's a natural way to move up and down in heterozygosity. But if I look within a set of within one of these colors, so within a particular population size, and I label all of those dots by their observed heterozygosity, the more heterozygosity observed, the more likely there is that coevolution is still occurring in that population. And the less heterozygosity I observe, the more likely I am to observe directional selection in that system. All right. So what does this mean in conclusion? To answer our question, does interspecific negative frequency dependent selection maintain genetic variation? No,
It doesn't. In fact, the neutral stability of those cycles makes matching alleles coevolution behave largely neutrally, but we see these small deviations above and below that neutral expectation due to the directional selection once a parasite type fixes. Okay, so variation can be maintained transiently, but will not be maintained in the long term. All right, so this is a talk on the ecological maintenance of genetic variation, and we know that coevolution does not occur in the absence of other ecological factors. In particular, we know that infection of hosts by parasites leads to host death, and so we should include these Locke-Volterra style host parasite dynamics in the model if we truly want to understand what the impact of coevolution is on the maintenance of variation. So here I have now just slightly modified the model we had earlier, but now instead of keeping the host and the parasite population sizes constant with this Moran style model, I have host birth events and host death events, and similarly parasite birth and death events that allow the population sizes to change over time. What do we see in this case? Well, let's first look at the limit of infinite population size. Here we see those fluctuations in the allele frequencies, but now they're slightly cycling in. So they slowly cycle in toward this interior point. And this is a particularly slippery type of cycle to get your hands on because it's not, if I were to look at those leading eigenvalue for this internal equilibrium, it would still have a zero real part, but it turns out it has a second order term that is stable. So these are slightly stable cycles, or second order stable cycles. And we can see this when we look at that average heterozygosity over time, where we see these fluctuations in heterozygosity. But now if you average that heterozygosity over time, it approaches 0.5, or the maximum value. All right, so there's already distinct differences in the deterministic model. How does this play out across parameter space? So this is a similar type of model with expected versus observed heterozygosity. And we see a remarkably different pattern where we now have an on average maintenance of genetic variation. So what determines the variability amongst these points within a particular population size? Well, the same processes that we had before. If you end up really low in this cluster, that might be because your pathogen fixed and there was directional selection. There can be the relative rates of host and parasite turnover. But in addition to this, we now have this factor of stability. So the more stable those slightly stable cycles are, the faster you'll cycle in toward that intermediate allele frequency, that polymorphic equilibrium, and the more genetic variation will be maintained in the long term. So there's no direct analytical expression for this stability, but what you can do is you can go back to these cycles here and you can fit them with a sine wave that has a decreasing exponential in it, and then you can use the rate at which those exponential cycles decay, or that exponential decay, to estimate the rate of second order stability. All right. So this is not the only way we could have put ecology into this model. In this world that we live in now, this is not going to be a surprise, but another way that parasites can inhabit hosts is that they can be transmitted directly from one host to another. So what if instead of modeling this in this free living parasite that bumps into hosts within an environment, we model the direct transmission of a pathogen from one host to another? So in this case, I have two different susceptible host types, and I have four different types of infected hosts. So two different, depending on the host genotype and the parasite genotype. And then I have three processes. I have infection, parasite death, which leads to the loss of an infection that becomes susceptible again, and then host death and parasite death. All right. So what happens in this model? You've now seen this kind of procedure we look at. We first look at what happens with the deterministic dynamics, and it turns out that this result is kind of interesting in and of itself. So the traditional thing that determines the stability of an epidemiological model like the one we just presented is the basic reproductive rate. But now we have a coevolutionary epidemiological model. So this basic reproductive ratio, R naught, is now going to depend on the frequency of the host and the parasite. So a way to visualize this is to look at the matching transmission rate. So this is the rate at which matching hosts are transmitted, or matching parasites are transmitted to matching hosts. And on the y-axis, the mismatching transmission rate. So this red line here is that classic R naught equals 1. And so if you fall above that red line, the disease is going to spread and there's going to be coevolution. And if it falls below that line, then the disease will ultimately go extinct. But how the disease goes extinct or manages to survive is a little bit more complicated. So if you live in this dark purple area, then the parasite is guaranteed to go extinct, is unable to spread regardless of coevolution. In this light pink area, it's actually coevolution in the change in allele frequencies that drives the parasite extinct. In the light green area, coevolution now starts to cycle and actually is able to pull the transmission rate up and enable the pathogen to spread. And then finally, in this dark green area, the disease will spread regardless of coevolution. Okay. But independent whether we fall into this dark green area or this light green area, the thing that we observe is stable cycles. So this internal equilibrium is now truly stable. It's not second order stable, it's linearly stable. So there's actually a little region here that you don't observe cycles in at all, but what it requires is like super uneven transmission rates between matching and mismatching hosts, and really high transmission rates, which kind of seem out of the realm of what we would observe in the real world. So we'll focus on this cycling dynamic today. All right, so that's the deterministic dynamics. What happens at the stochastic level? Well, you've seen lots of these types of plots before, and now we see even a different dynamic where when the population size is small, there's a net decrease in heterozygosity, and when population sizes are large, there's an actual substantial increase in heterozygosity relative to the neutral expectation, which is this dashed line. So in particular, there's some of these points that fall way up here with substantial maintenance of heterozygosity. All right, why do I land somewhere in this plot and not somewhere else? Well, you've seen these before. It's the same three processes. One of them is the process of pathogen fixation. The other one is the relative turnover rate in the host and the parasite. Now, in the previous two models, we could change host and parasite death rates and turnover completely independently of one another because the host and the parasite were living separately. But when you have a directly transmitted disease, you can't have like a infected host die without the parasite that it infects dying as well. So now you can't completely change these two things independently. In particular, you're forced to have parasites have shorter generation times than the host, but to what extent we can do that, that matters. Finally, there's a stability analysis where you have this leading eigenvalue lambda that tells you how fast you're approaching this polymorphic equilibrium. And the more stable that polymorphic equilibrium is, the more likely you are to maintain heterozygosity and fall into one of these points up here in parameter space. All right, so there's been a lot of models here, so let's just go over what we have seen. We had a matching alleles model of host-parasite coevolution that had no ecological dynamics in it. And in this case, the host and the parasite are only coupled through the action of reciprocal natural selection. In the intermediate model, we have both this reciprocal natural selection and demographic dynamics that are created by by parasitism. And then in the last model, the host and the parasite are actually coupled in three ways. They're coupled through reciprocal natural selection, demographic changes, and the incidence of the population. Okay, so in this first case, we saw neutrally stable cycles, second order cycles, and linearly stable cycles. cycles. So the more and more coupling we have between the host and the parasite, the more and more stable this internal equilibrium becomes. And then this plays out on a larger scale when we look at the maintenance of genetic diversity, where with only neutrally stable cycles, you don't maintain genetic variation. In fact, you deplete genetic variation. In the middle case, you maintain small amounts of genetic variation. And in the last case, it's super variable, but in some parts of parameter space, you can actually maintain substantial amounts of genetic variation. So I've been throwing out these words like not very much variation, moderate amounts of variation, and a lot of variation. And what do I mean by that? Well, we can compare them to these gray lines, which represent weak levels of overdominance, medium levels of overdominance, and loss of overdominant selection to get an idea of how much variation is being maintained in this model compared to other processes that are known to maintain variation. So only in the case where you have epidemiological dynamics are you likely to have points that are consistent with having very strong overdominant selection. All right, so in conclusion, despite long-standing hypotheses about the impacts of coevolution, negative frequency-dependent selection that interacts between species does not, in fact, maintain genetic variation. Matching allele coevolution actually creates the same kind of processes that deplete genetic variation once a parasite type fixes. It can lead to directional selection and the net depletion of genetic variation. We can somewhat rescue variation by including ecological or epidemiological feedbacks, but this is only able to maintain levels of genetic variation seen under weak overdominance or in a very few parts of parameter space in the epidemiological model, more variation. So another way to think about the maintenance of variation is we're only going to see coevolution currently happening in a population if both host types are around and both parasite types are around, which requires the maintenance of variation. So under what conditions are we most likely to observe ongoing coevolution in the natural world? Well, we're most likely to see it in this small region in parameter space in an epidemiological context. So these are the evolutionary conclusions from this talk. But this set of models taught me a lot about evolutionary modeling in general. And so I wanted to share with you some of the larger conclusions that I learned about modeling ecological dynamics. So first, it was really necessary to make progress in this in order to make progress to specify a specific quantitative hypothesis that compared evolutionary dynamics to some neutral expectation. So long story short, Kamura is right. We need neutral models. And the matching alleles model is particularly sensitive to model assumptions because of the neutral stability of this polymorphic equilibrium. And finally, even though we were interested in the stochastic dynamics with genetic drift, by looking at the deterministic model and looking at these eigenvalues, it was remarkably good at informing us on what we should expect in the stochastic case. So deterministic models can give you insights into more general model dynamics that include neutral genetic drift. And so those are my conclusions, and I'd like to thank you for letting me present.
Thanks a lot for a great talk. So are there, I don't see questions on Zoom, but are there questions in the real life version?
There are.
Okay.
There is one.
Hi, Ailene. It's Leonard. Thank you for your talk. And so I was wondering, since you put like different size, like not constant size in your last model, how would your conclusion on maintaining genetic variation will still hold if you add like spatial structure and migration in with your epidemiological dynamics?
Did I plant you in the audience? I was really hoping somebody was going to ask a question like this. So there's like, this is all well and good that matching alleles models don't actually maintain genetic variation, but there's a deep conflict here that we can build models of coevolution in spatially structured environments and observe local adaptation. And local adaptation requires genetic variation. So how can we not maintain genetic variation within single populations, but then observe local adaptation? What does migration do to fundamentally change coevolution to maintain variation? And I have no good answer for you. I was hoping to actually include this in this talk, but I'm working on a model with a student at the University of Toronto to study this, and it's particularly unwieldy. So I will have to get back to you on what happens there. Unfortunately, I didn't get it straightened out enough to tell you.
Fair enough. Thanks.
Okay, then I also have maybe a quick question. So in your talk, you gradually removed some of the artificialities of the first model, which didn't have demography in it, and then you included it. But then also the matching allele model is very is specific in that there is a lot of symmetry in it. And is it also something that, so you kind of mentioned it already in the introduction, but is it something that you're also looking into different, if you remove this symmetry and also add dimensions instead of having two, I don't see you, so sorry, it's confusing on Zoom. Only see myself.
Of multiple species or dimensions as far as multiple loci?
Both.
Okay, so first of all, let's talk about the matching alleles model. So there's this great paper by Neil et al. That studies like the continuum between a matching alleles model that's super symmetric and this gene for gene model that is the opposite of that. And what they find is that it doesn't take a whole lot of deviation from this symmetricness to become very gene for gene like dynamics. And so the loss of symmetry definitely has impacts, but the thing is as soon as you turn into a gene for gene mode, then your null expectation is actually not to maintain variation because the gene for gene model favors fixation. So then you're breaking the maintenance of variation in another way. But so I haven't thought about exploring that too much. But because I don't expect it to maintain variation to begin with. Mm-hmm. As far as adding more species or more loci, I haven't thought too much about adding more species, but adding more loci would be really fun because even in these simple cases or most well-studied cases like the Daphnia and Pasteria, we know that there are multiple loci involved and that there are epistatic interactions between those loci, even in these classic model systems of matching alleles models. So I definitely think that building that element of realism in there and seeing if epistasis between matching alleles loci could maintain variation would be really interesting.